In our previous video, we have already discussed the initiation of DNA replication process taking place in the prokaryotes, and also we have discussed the elongation process of the same, where we have already seen that how smartly the DNA polymerase do incorporate nucleotides at a time, thereby elongating the newly synthesized stretch of DNA. Now here in this lecture, we will discuss about the termination of the DNA replication process and the E.Coli. And at the end of the lecture, we will have the two newly synthesized E.Coli cells within which the genetic material which has recently been synthesized will be incorporated. Now, scientifically it is proved that for the replication of the genetic material, it takes 40 minutes at 37 degrees Celsius. And it's pretty obvious that it's only after the replication of the genetic material that the cell will undergo division. But why I'm saying this? I'm saying this because scientifically it is also proved that an E. coli cell takes only 20 minutes to divide. Now, how is it possible? Where on one hand the genetic material takes 40 minutes for its replication process, and on the other hand the cell takes only half the time, that is only 20 minutes at 37 degrees Celsius, in order to form two new daughter cells. Now, if you think about it, it can only be possible at only one condition, and the condition is that even before the first round of replication is completed, the second round of replication from the two newly synthesized double-stranded DNA has already begun in such a way that at the time the first round of replication is completed, has reached its end point, the newly synthesized double-stranded DNA have also completed half, that is 50% of its replication process. So, doing such kind of mechanism, what is the benefit for the E. coli? The benefit is when this 50% already replicated genomic material would be incorporated in the two new daughter cells. Then the two new daughter cells, whenever it would opt for the replication process, then it would only have 50% of the job that is to be done, so thereby saving half the time. In order to understand about this and also about the theta replication, which is the end product of this mechanism, which I have already discussed right now in this particular video, you need to stay tuned with my lectures. So without any further delay, let's begin our lectures. Thank you very much.
So let's begin our analysis of the termination process taking place in the prokaryotes. So as we know that bacterial genomes are replicated bidirectionally from a single point or a region known as the RIC, which means that the two replication forks should meet at a region opposite to the RIC. Yes, definitely. Now we know that the process of replication will begin from here, which is known as the RIC. Now with the initiation of the process of replication, there will be formation of two different replication forks, and the two different replication forks would move opposite to each other in this way. Now see, this is the first replication fork. You can see here, this is the second replication fork. Now if this replication fork continues its movement, it will meet somewhere at the opposite end of the RIC, right? Now in the region which is opposite to RIC, the replication of the genome terminates, and yes, it is in this region which is present opposite to the RIC that the process of replication terminates. Now how it terminates, for that look at a diagram. Now as you can see in the diagram, which is a very schematic diagram of a circular DNA of the E. coli, here I have marked certain regions as red. These red marked regions are actually certain repetitive sequences which is made up of approximately 23 base pairs. Now these repetitive sequences are known as the ter sequences or the terminus sequences. And we have got 7 such units of repetitive sequences. Now the next question comes that what is the importance of these repetitive sequences? These repetitive sequences act as a binding site for a very special protein known as a TUS protein. This TUS protein, after associating with the ter sequences, exhibits the process of termination of the DNA replication. How? For that, let's look at this particular diagram. Now as you can see the replication fork moving bidirectionally, and as I have said, this is the first replication fork, this is the second replication fork. In order to erase your confusion, I would like to say that this DNA strand, which you can see, this is the first parental strand, and this DNA strand, which you can see, it is the second parental strand. And these strands that you can see, which I am marking it as dot, this strand is the newly synthesized strand. Similarly, this strand is the newly synthesized strand. Now, in a newly synthesized strand, you can see that there is a strand which is continuous, and there is another strand which is represented by dotted lines. Now the strand which is continuous is the leading strand, and the strand which is discontinuous, it is the lagging strand. Hope the confusion at this region is cleared. And a similar thing is exhibited here in this direction also. These are a parental strand, this and this, and these are the newly synthesized strand. Now you can see the strand which acted as a leading strand at one side of the replication fork, this same strand is acting as a lagging strand at the other side of the replication fork. Now this thing has already been described in my previous video. If you have not checked it out, and if you have confusions regarding this, please go and check it out and then come to this particular video. Now, since it is clear that which is the leading strand, which is the lagging strand, how the replication fork is moving and so on, now the next thing which you need to understand is this TUS protein, after associating with the TUS sequences, it will permit the movement of the replication fork in one direction, but will inhibit the movement of the replication fork in the other direction around. Now this would depend on the orientation of the TUR and the TUS protein. Say for example, here you can see I have written clockwise trap, which means if a replication fork is moving in a clockwise direction, then it would be trapped. It would not be allowed to move above this termination site. Similarly, I have written here anti-clockwise trap, which means if there is a replication fork which is moving in an anti-clockwise direction, then it would be trapped here. Okay, now having understood this, let's look at this particular thing. Now as you can see, the replication fork is moving in an anti-clockwise direction. Yes, it is moving in an anti-clockwise direction. So since it is a clockwise trap, therefore it will permit the movement of anti-clockwise fork, and the fork would move till here. But this particular region is an anti-clockwise trap, so therefore the anti-clockwise moving fork would not be allowed to move ahead of this particular region. Therefore this particular replication fork would be trapped out here in this region. Similarly, the fork which is moving in this direction, that is in clockwise direction, would be easily be able to pass through this trap, because this trap is not for clockwise direction moving fork, it is only for the anti-clockwise direction moving fork. So the fork which is moving in this direction can easily cross this trap, but it will be unable to cross this particular trap, as this particular trap is a clockwise trap, and it will not allow the movement of any replication fork which is moving in a clockwise fashion. So therefore both the replication fork would be trapped out here, and this would mark the end of the replication process. Now after the replication process ends here in this particular region, these two newly synthesized double-stranded DNA would be interlocked with each other. This interlocked DNA would be separated from each other by the help of a topoisomerase, which is known as topoisomerase 4, by a type of topoisomerase 4. And we know that topoisomerase has got an ability to crack open between the double-stranded DNA and separate this two circular DNA. So that's it for termination. Hope you have understood the process of termination very clearly. Thank you very much. Let's analyze another beautiful phenomenon that occurs during the replication of the prokaryotes, more specifically in the E. coli, that is, the theta replication in E. coli. As stated earlier, fully methylated condition of the double-stranded DNA or IC is required for the initiation of the replication. What I do mean by fully methylated condition of the double-stranded DNA or IC is, within the ORIC, there are various repetitive sequences present. Of the many repetitive sequences, one of the repetitive sequences is GATC sequences. Now this GATC sequences is aligned from 5' to 3' direction within the ORIC. Now in the GATC sequences, as you can see, there is an adenine residue. Now in all the GATC units present within the ORIC, this adenine residue must be methylated for the replication process to move forward. If any of the adenine residue belonging to the GATC sequences present within the ORIC is not methylated, then replication will not proceed forward. Here, one thing is very much clear, that if replication needs to occur, then methylation of the adenine residue in all the GATC sequences present within the ORIC is a must. Having understood this, let's suppose all the GATC sequences are methylated, and a methylation of the GATC sequences is demarcated by these red dots. Once methylated, now the double-stranded DNA will go for replication. Now, as the replication is initiated, there will be the formation of two replication forks, one at this end and the other at this end. Now, with the initiation of the replication fork, various enzymes will join in and move forward the process of replication, and initially it would replicate the ORIC region. When the replication of the ORIC region is accomplished, there is a formation of two newly synthesized DNA which contains the ORIC region. For example, this is the first newly synthesized DNA which contains the ORIC region. This is the second newly synthesized DNA which contains the ORIC region, and these two strands, where you can see this red dotted picture, is the parental strand. Okay, having understood it, as it is depicted in the diagram, that these red dots are not represented in this newly synthesized DNA, which does mean that this newly synthesized DNA or IC is not methylated. That means this double-stranded DNA is in a hemi-methylated condition where the parental strand is methylated, but the newly formed DNA is not methylated. Now in this hemi-methylated condition, there is a protein known as Sec-A. This protein binds to this newly synthesized DNA. Now when this protein binds to a newly synthesized DNA, it inhibits the action of DNA A protein, which is very essential for the replication process to begin. On the other hand, it also inhibits the methylation process in the ORIC region. Therefore, immediate reinitiation of the replication from this newly synthesized DNA does not occur. But the fact is that this Sec-A cannot inhibit the methylation of the newly synthesized DNA for a very long time. After some time, this Sec-A protein gets dissociated, and once dissociated, this newly synthesized DNA now is no more in a hemi-methylated condition, as methylation occurs with the help of an enzyme known as dimethyltransferase, and also there is an association of DNA A protein, which is very much important for the initiation of the melting process to occur. Now as the prerequisites are taken care of, now the replication also begins from this newly synthesized DNA even before the first round of replication is completed in this particular fashion. Now here you can see that even before the first round of replication, now see, this is the replication fork, right? This replication fork is not yet terminated, and at that particular time there is an initiation of replication from the newly synthesized double-stranded DNA with the formation of 6 replication fork. Now you can see that 2 replication fork was already formed in the previously being executed replication. Now with the initiation of replication in the newly synthesized DNA, another 4 replication forks are formed here is the second, here is the third and fourth and fifth replication. So on a total there are 6 replication fork which is formed. So what we have understood from here, that even without the completion of first round of replication in the E. coli's chromosomal DNA, the second round of replication within the newly synthesized DNA occurs. So therefore, whenever the first round of replication is terminated and this two newly synthesized DNA is separated with the help of topoisomerase 4, this circular chromosomal DNA looks like this. Now you can see this is the half replicated DNA, that is, even before the first round of replication was completed, half of the replication, that is 50% replication of the newly synthesized DNA was already completed. And therefore, when the circular DNA is separated from each other by the action of topoisomerase 4, we get the two new daughter chromosomal DNA in this particular shape, which acquires a theta shape, and therefore the mode of replication where the chromosome acquires a theta shape is known as theta replication. Now as you can see here, that in this theta shaped chromosomal DNA, half of the DNA has already been replicated, and therefore whenever now this particular chromosome feels the requirement of going for replication, it will complete the replication process in 50% less time, because 50% of the replication was already completed when the first round of replication was being initiated. So therefore it will require 50% less time to divide and form two new daughter cells. Therefore it is said that for the division or for the replication of the chromosomal DNA of the E. coli, it takes 40 minutes, but the cell division takes only 20 minutes. It is because during the first round of replication, the second round of replication of the newly synthesized DNA begins, because of which the newly synthesized chromosomal DNA acquires a theta shaped structure, and also since 50% of the DNA replication is already accomplished, so whenever the new daughter cells go for the replication process, it will take 50% less time in order to execute the replication process, and thereby would follow a cell division process. And therefore it is said that for the replication process it takes 40 minutes, but for the cell division process it only takes 20 minutes, because 50% of the DNA replication has already been completed in the previous stage itself, even before the completion of the first round of replication. I hope this concept is understood by you guys. Now if you have loved this concept and the content in this video, please hit the like button and subscribe to this channel. Thank you very much.